course with some other lecturers, professors from the Department of Medical Genetics. Do you know anything about genetics?
Some general information. Nothing
No,
much.
nothing.
No, just some general information, nothing specific.
General.
Yeah.
What about you?
The study of genes and heredity. Study of genes and heredity.
Study about the traits which are transferred from parents to their
Heredity.
And you?
Mutations that occur.
You?
Just about the genetics.
Just about genetics.
So we get to
Very
know
good.
answers, right?
In this course, we are going to talk about basic and modern concept of genetics, with actually emphasizing on medically relevant materials. This will prepare you for to understand genetic contribution of really some conditions that may cause some disorders and the way how we can treat them in the future. Actually, we expect to attend to all classes. The administrative structure for this course, as I said, is for the Department of Medical Genetics, course director, and also other teaching faculty members. We will have exam at the end of the course. This will be an MCQ exam, and we will test your knowledge about the objects that we will talk about them. If you have any question, any problem, you can email me and or other professors who will teach you. This is your schedule for this semester. Today, actually, we will have 3 classes. Number 1, 2, and 3. What? I cannot hear you.
We were talking about the time of today's class. It's up to 3, right?
Up to 3 years.
Today.
Yes. We will talk about an introduction to medical genetics, mode of inheritance, molecular genetics, and genetic counseling and pedigree analysis. And tomorrow, number 3 and number 4 and 5. And maybe by 1 or 2 months later, other professors will talk about other subjects. We actually designed to provide some material which will help you to understand the genetics principles. As I said, we will talk about genetic containing structure in the future, and also at the end of class you will be able to use genetic tools in clinic and maybe some research purposes. By the end of this course, you should be able to describe what are genes, and how, maybe at the top of the slide, what are genes, how they are organized, and how they segregate and inherited from their parents. We talk about the organization, replication, and transmission of the chromosome. We will talk about the nature of mutation and their contribution to some genetic disorders and variability. We will talk about the pattern of inheritance, autosomal dominant, autosomal recessive, and X-linked inheritance, and also about mitochondrial inheritance. We talk about genetic imprinting, multifactorial disorders or complex genetic disorders, how to perform risk factor calculation for Mendelian inheritance. We talk about the congenital abnormalities and approaches to diagnose. We talk about common molecular and cytogenetic diagnoses, and we talk about PGD, perinatal genetic diagnosis, and also PND, perinatal genetic diagnosis, and brain plantation genetic diagnosis. And we talk about genetic counseling, and also we talk about some approaches to treat genetic disorders. Our reference is essential medical genetics, MA Government of Medical Genetics, and also lectures. If you have any question about the course, you can ask me right now. DNA, some definitions, and also terminology in medical genetics. You know about the scientist who discovered DNA? Who was the scientist who discovered DNA? Okay. You know Mendel? Have you
Yeah.
heard about Mendel?
Yes.
Who is he? What did
He
he
was
do?
a man official who like started the foundation for the genetics. He used like pea plant.
Yeah.
Mostly.
9 threads.
Yeah.
We study about 9 threads.
Seven.
Seven
Cells.
Okay. Actually, he is known as the father of genetics. He was discovered the rule that genetic trait is transmitted from parents, and he was an Australian born in 1822. Maybe after his death, his knowledge was pay attention to his knowledge in 1900. As I said, he is known as father of genetics. What about these two scientists?
They
What?
discovered.
They gave the model
Basis
for
of
DNA.
DNA. Let's show the basis.
Actually, they discovered the structure of the DNA. The first scientist who created a model of DNA. This model is still used, and also, as you know, DNA is made of sugar, phosphate glucose, and they run as a ladder, and by nitrogen bonds. And there are 5 elements in the structure of DNA, which is carbon, hydrogen, oxygen, and nitrogen, and phosphorus. What is DNA? What?
I said it's the basic unit of health.
I cannot hear.
It's the basic unit of health.
It's
Basic. Like the
Basic?
Yeah. Functional unit.
when we talk about the inheritance, actually, there must must be something that keep our information and transmit this information to the next generation. Okay. The basic of the physical and functional of heredity is DNA. Okay. DNA is necessary for synthesis of some functional protein and RNA molecule. Actually, any traits that we have, like hair color, eye color, palms, or any information that we have is carried out by DNA and is transmitted to next generation. Okay. DNA during the transcription, there are two steps for actual production of the protein. The first one is transcription. The next one is translation. Okay. When DNA needs to be actually transcribed, what does transcription mean?
Making DNA from
Transfer the information. Like from
Copying mRNA,
messenger RNA from DNA.
copying mRNA from DNA. We call them as transcription. And at the beginning, we will have primary transcript, and then we will have some processing on RNA. It's called as RNA processing. Then we will have mature mRNA, and then this mRNA will be transferred from nucleus to cytoplasm, and then we will have translation. And what does translation mean?
And they make a protein out of an mRNA.
Making polypeptide from mRNA. Okay, and this polypeptide will have some processing, and then we will have active protein. This active protein will have some function in our body, and then we will be degraded. Okay? Any disease or any disorders that we have will happen. Actually, some changes will happen because of some changes in this actually way. Okay? Each part of this way, that if you have some problems, some changes, then we have some genetic disorders. It depends on the what gene is affected, what protein is coded by these genes, and what function this protein have. Then we will have some phenotype or some changes in our normal function.
Doctor, where can we find this is a process, and where can we find this is a process? And
Where can we find?
Yeah.
It's the result of maybe 100 years of study. Many scientists work in this field, and they found out, okay, this is the way that we will have protein from DNA. Okay. What do you think this slide will talk?
It's a comparison. It's a comparison.
Comparison now?
Going
Yeah,
from
yeah.
very basic to complex. From very basic to all complex.
It's a comparison about our molecular things that are going place. For example, in encyclopedia, we are talking about, we are making a comparison with the chromosome set and the single book, which is the volume, and then we are making it the comparison to a
Actually,
chromosome.
it's compressing the information because first, like there was a large book, then they made it small, and it was just a page.
Actually, this is like when we say our information is stored or transmitted by our DNA, is something like this, something like encyclopedia. Encyclopedia included some information about the subject. Okay. If this encyclopedia has 23 volumes, it's something like the number of our chromosome. Okay. We have 23 pages of the chromosome, is something like an encyclopedia, which has is something like an encyclopedia which has 23 volumes. Each volume included some information. Okay, it's something like each chromosome has included somehow our information. Okay, all these volumes are related to each other. Okay, if you do not have, for example, volume number 1 or volume number 10, maybe this will not be complete. This information will not be completed. Okay?
It's not that.
Yeah. If you lost one part of your chromosome, it's something like you lost a volume of this encyclopedia, then the information will not be complete. Okay? Maybe volume 1, there are some subjects in volume 1 which is related to volume 10. Okay? If you want to understand what information you need in volume 10, maybe you need know about the volume 1. If you do not know that information, maybe you do not have function on volume 10. You do not understand about the volume 10. Our information is something like this. We said 23 chromosomes. Each chromosome included some of our information. And this information are related to each other. Okay, maybe some of our information is included in volume 9, and other part of related information is included in volume number 10. You need to have both volumes to have complete information. So each volume of this encyclopedia is like a page of the chromosome which included some information. Okay. When you open this book, each page of this book, maybe it contains a few genes. Some information is in previous page, and some information may be included in next page. You need to have all the pages together to have complete information. If you select one word, it's something like genetic code, okay, which has 3 actually alphabets, and then when you make a sentences or select a sentence in one page, it's something like select a gene which included several codes, and this code actually is specialized for a protein. So the complete sentences is something like a functional protein in our body. As I said, all these volumes are related to each other. All these pages are related to each other. Okay. All these sentences is related to each other. Maybe you need to know all information. You need to have all actual volumes to have complete information about this encyclopedia. When you select one part of a chromosome, each chromosome actually included 22 to 48 to the power, multiplied by 10 to the power of 6 nucleotide pairs of DNA. If you select about 10% of these chromosomes, maybe it included about 40 genes. When you actually select about 1% of each chromosome, maybe it included about 4 genes, and each genes have different actually parts we call them as regulatory region. You know what's the regulatory region?
Has some enzymes. Has some enzymes.
Enzymes.
Yeah.
Its name is clear. Okay, it's a
Basically-
regulatory region. It means it's regulate.
The gene or the
It regulates the expression of the gene.
Expression. DNA formation.
Regulate the expression of the gene. What does expression mean?
The transcription, how much part of
How this gene is producing the protein. Its regulation is regulated by regulatory region. There is a DNA sequence, usually is located just before of the starting structure of gene. This regulates at what time, at what tissue this gene must be expressed, or this protein must be synthesized. Okay? In which part of our body, what protein we need, and at what time we need that protein. Okay? This is regulated by regulatory region. Okay? And also, there are other parts in each gene. We call them as the exon and intron. What are exon and intron?
I think it regulates it. The endonucleosome.
But
Endron like cuts the DNA in from inside, but the exon it cuts it on sides.
I think the intron, the space between the exon.
yes, and what are they doing? And exon is the space between introns.
exons are excluded from the
Exons are excluded.
excluded, I think, yes.
And
Then introns are
how
spliced.
exons, the process starts.
During splicing process, exons are excluded.
Exons are excluded. Maybe.
I forget.
There are two parts in each genes. Beside the regulatory region, we call them as exon and intron. Exons are coding region, okay, and intron are non-coding region, okay. When RNA is copying from a DNA, we will have both exons and intron which are copied, and then intron are cut and exon are joined to each other. Then we will have just coding region. Okay? So this coding region actually is the functional part of a gene. But there are some spaces between the exons that we call them as intron, but they are not. They are included in the structure of the gene, but they are not included in coding the amino acids. Okay, they are deleted when RNA is processed. And then when we talk about the gene expression, it means making protein from DNA. Okay? Protein is made, protein has some function in our body. We have processing on protein, and then functional protein will have some function in our body, and then will be degraded, functional protein. Okay? Any question? We are going to talk about some definitions and terminology in genetics. Maybe you know some of them, but maybe you don't know most of them. You know anything about gene locus and allele?
Locus that means fox. Locus. That means it's called fox.
Fox.
Yes. remember locus coeruleus.
Focus over. It's basically the address of a gene, of an allele on a gene, right?
The address,
Like, yeah, sort of.
the location of the gene on
On the
each
chromosome.
chromosome,
Yeah.
the position of the gene on each chromosome. As we said, we have 23 pairs of the chromosome. Okay, one gene may be located on chromosome 1 and short arm of the chromosome 1, or long arm of the chromosome 1, and which part of this long actually arm or short arm. There are specific address for each gene. Okay, when we want to talk the address, when we want to talk about a gene, we need to know about the location of that gene on each chromosome, which arm, which part of this arm, which location of this actually arm of the chromosome this gene is located. We call them as locus. Okay. A specific position of each gene on each chromosome. We call them as a locus for each gene. What is the allele?
Or gene. It's a form of gene.
What?
Form of gene.
Form of genes. What does it mean?
It's a pair of genes on two chromosomes. They have the same locus. A pair of gene that is located on the same locus on both chromosomes.
On both chromosomes. We have two copies
Uh-huh.
of each chromosome. Okay.
Yes.
One comes from our father and one comes from our mother.
Mother.
In normal, both chromosomes actually has the same actually structure, and the same genes are located on each part of this chromosome. Okay, so it means we have two copies of each
Gene.
gene. We call them as alleles.
Alleles.
For each gene, we have how many alleles we have? 2 alleles. Okay. So for each gene, each individual has 2 alleles. But in a population, the number of alleles is not limited. Okay. Maybe for a gene, I have 2 alleles. You have 2 alleles different from me. You also 2 alleles different from me. 2, 4, 6, 8, 10, 12, 14, okay, 16, 18, 20. In this class, we have 20 alleles for a gene. Maybe some of these alleles are similar, some of them are different from each other. But the number of alleles in any population is not limited. But for each person, the number of alleles is just 2 alleles. Okay? What's the wild type allele for individual?
It says like it's a single allele when there's just, it has no other allele of it, like it's just one copy of a gene. Is that
What?
it? When there's only one copy of a gene,
Okay.
is that wild type? Because that's what's written there.
No, the normal form of each gene,
Uh-huh.
okay,
Uh-huh.
the normal form, which doesn't have any problem or any differences from the normal type. The normal form of each allele, we call them as wild type. Okay,
Yeah.
okay, for each individual, which is normal in any actually alleles, we call them as wild type. Any differences from the wild type, we call them as the variation. Okay.
Uncontrolled cell division. Uncontrolled cell division.
and
Uncontrolled cell division.
okay. And if we have some permanent changes in our DNA, we call them as a variation. This variation may be disease causing, or this variation may not be disease causing. Okay? Maybe for a gene, I have wild type 4, you have also two different alleles from me, but that one is also wild type, because it doesn't make any changes in the phenotype. He, she, you, and other, maybe we have different alleles, but all of them are wild type, because it doesn't make any changes in our phenotype. Okay? We call this variation maybe disease causing, as I said, or not disease causing. If it is disease causing, usually it's called as mutation. But not all mutation are disease causing. Okay? If this is not actually disease causing, we call them as polymorphisms. But some polymorphisms are also disease causing. Okay, it's a little bit complicated, right?
Can you repeat it?
Any variation from the wild type, okay, we call them as, any changes from the wild type, we call them as a variation. Okay? This variation may be disease causing, okay, or not disease causing. Okay? Usually, the disease causing variation is called as mutation. But not all mutation, all permanent changes in the DNA are disease causing. Okay? Any variation which is not disease causing, we call them as polymorphisms. But some polymorphisms are disease causing. Okay?
But how is it possible if we have disorder, or if we have any change in the, or as we say, variation, and we don't have any disorder? How is it possible?
It's possible because it depends on the in which part of the gene this variation happens. Okay? Some part of genes are not important. Okay? Because they code a protein, which that part of the protein is not very important. Okay? But some part of proteins are important. If any changes happen on that part of the protein, we will have phenotype. It depends on where these changes happen. This is important site for the gene and for the protein or not. If it's not important part of the protein, maybe we will not have any changes in the function of the protein, then we will not have any phenotype. What's the gene pool? Yeah, you can read it. The collection of
Available.
alleles in a population. I have 2 alleles for a gene. You have two. When actually collect all of them in a actual population, this is your class. Maybe we will have bigger population and bigger population. If we have all alleles, we call them as a gene pool for that genes. The distribution of each allele in each population is important. We will talk about this. For example, if we have a variation in a population which is disease causing, okay, and is common in that population, okay, maybe the frequency of the carrier will be high on that population. Carrier frequency will be high on that population. So the distribution of animals in the population also is important. What is this slide?
It's talking about genotypes and phenotypes.
What's the genotype and the phenotype?
Well, genotype is basically how, like, what type of gene expresses which thing. Like, different genes express different characteristics in a person or in a general being. But phenotype is the things that we see on, like, how do I say, outside, on the outside of the body, basically. It gives a difference in appearance.
Physical appearance of the phenotype is the physical appearance. Like Mendel study about this gene and this gene may relate to this.
Any other?
Genotype and phenotype mostly describes the variations, but here in the genotype we mainly talk about variation in the genetics, like we go deep down to the genetics, to the molecular level, while in the phenotype you can observe the trait or the variation, like physically on the outside, by the appearance.
When we talk about the genetic information of each gene for each individual, actually we talk about the genotype. Okay? Genotype may be talking about a part of a chromosome or just a nucleotide of a DNA. Okay? When we talk about the structure of DNA for each gene in each individual, actually we talk about the genotype. Okay? When we talk about the appearance or the expression of that gene, how this gene is coding a protein, what function that protein have, and what appearance we will have based on the function of the protein. We are talking about phenotype. Okay? Genotype is actually something like music note. Okay? It's something like music note. If you understand this music note, you know each note coded for a voice. Okay, for a part of this, each part of this note is coding a kind of the actually noise of the music. When you start to play, this note is something like your phenotype. Okay, what you hear from this note is the phenotype. Okay. Our genotype is something like music notes. What we hear from this music note is something like our phenotype. Okay? What's the relation between the genotype and the phenotype? When we talk about the genotype and the phenotype, we will have some actually problems, or we will have some actually difficulties with some phenotypes. We have a term, we call it as genetic heterogeneity. What does it mean?
Didn't hear.
Have you heard about genetic heterogeneity?
No.
No. Sometimes we have actually similar phenotype from different genotype. Okay? Similar phenotype from different genotype. We call them as genetic heterogeneity. Different genotype make the same phenotype. For example, for some genetic disorder like Down syndromic hearing loss, or cystic fibrosis, or Alzheimer, or autism. You have different genotype. It means different mutation, different changes in different genes causes the same phenotype. Okay. For example, for hearing loss, you have more than 130 different genotypes, different genes. Each genes that have some changes, the phenotype will be same, hearing loss. Okay. Different genotype causes the same phenotype. Or for cystic fibrosis, or for some other disorders like Alzheimer, autism. Different mutation in different position of a gene, or different mutation in different gene. They all have the same phenotype, which is hearing loss, for example. Okay, we call them as genetic heterogeneity. You got it?
Yes.
Also, for genetic heterogeneity, we have two types of genetic heterogeneity. One is allelic heterogeneity, and the other one is locus heterogeneity. What are these two types? You can read and explain. What's the allelic heterogeneity?
Mutations I've seen. More than 200.
What?
More than 200 mutations in a gene.
As I said, each gene has several exons. Okay? Exon, for example, 1, 2, 3, 4, 5, which are coding regions, which code a protein. Okay? If we have different mutation on the same gene, for example, one individual has in exon 1 individual has mutation in exon 10, one individual has mutation in exon 11, one individual mutation in exon 9. Okay? They are different mutations, but on the same gene. Okay? We call them as allelic heterogeneity. Okay? Sometimes we have locus heterogeneity. Other locus heterogeneity?
The mutations are occurring at different loci. For example, in Alzheimer's, mutation is occurring in APP, PS1, PS2, and APOE4. Yeah. And
This
just
is a code, Doctor. This is a code you can find in one exon. APV, PS, and you can find it in just one exon.
No, in different gene, different locus.
Okay, different.
Okay? As I said, for example, for hearing loss, we will have more than 130 genes that are related to the hearing loss. Okay? If one individual has mutation on a gene which is located in chromosome 1, he or she will have hearing loss. Okay? The phenotype is hearing loss. Another individual has mutation on another gene which is located on chromosome 9. Okay? This gene is related to hearing. Okay? If mutation happen on this gene which is located on chromosome 9, he or she will have hearing loss, but on different chromosomes. One is chromosome 1, the other one is on chromosome 9. Both are related to hearing, and the phenotype is hearing loss. Okay? So if we have mutation on different genes, on different locus, we will have locus heterogeneity. But if we have different mutation on the same gene, but different part of this gene, exon 1, 2, 3, 4, we will have the same phenotype. We call them as allelic heterogeneity. What's the phenotypic heterogeneity?
Mutation in the shape. Shape mutation.
Shape.
I mean form. The form.
We have different, in phenotypic heterogeneity, we have a different mutation
For example, eye
of
color.
the same gene, what causes different phenotype. Okay? For example, one individual may have mutation in exon 1. He or she will have some phenotypes. Another individual has mutation on the same gene, for example, on exon 3. He or she will have different phenotype. Different mutation on the same gene. What causes different
Phenotype.
phenotype? We call them as
Phenotypic heterogeneity.
phenotypic heterogeneity. Any question? Question?
Can you give us some more examples about phenotypic heterogeneity?
Like Gaucher disorder or neurofibromatosis. One individual, as I said, has mutation on exon 1. Is he or she will have, for example, neurofibromatosis type 1. It has some characteristic in phenotype. Another individual has mutation on exon 3. He or she will have neurofibromatosis type 3, which has different actually characteristics.
But it cannot occur in the same person. Like say in me, the same gene will get mutated. It's not possible.
It's not the same
It's
genotype.
not
Different.
for the same individual.
What?
It's not for a like single individual. The same gene will get
The same in different individual.
Yeah, different.
Different individual, yeah. The same. One individual cannot have two different phenotypes. Okay? One individual cannot have two different phenotypes.
Does eye color differences like also gets included in that? The difference in eye color is also included in that or not?
Yeah, maybe.
Like
different
let's say I
phenotype.
have blue eyes, you have brown eyes, that thing. Because they might have the same gene
The
on
same gene but different
but
changes.
different changes, right?
Different changes.
But
What to do in two different individuals, not the same individual. Okay? Mutation on exon 1 happened, for example, in my genotype. Mutation on exon 3 happened in your genotype. You have different phenotype. I will have different phenotype. But same gene, different part of the same gene. You know about the zygosity? Yeah? No?
I know.
As we said, we have two copies for each gene. Okay? If these two copies are the same, we call them as homozygous. If we have two copies which are different from each other, different means very small changes between them, but the function will not cause any disorders. Okay, all of them are normal, okay, but different from each other. We call them as heterozygous. Okay, and we also have compound heterozygote and double heterozygote. What are these? Compound and double heterozygote?
Compound heterozygous might mean that two different, like more than one gene, like might have different alleles. Or one locus might have different alleles on a single,
Single gene.
yeah,
Two different, for example,
genes.
one individual has two different, actually,
Alleles on the same locus.
alleles on the same locus.
Yes.
For example, he or she is heterozygote for one gene, one part of
One
the
part
gene,
of the gene, yes. Plus
and
one more.
other part of the gene. Okay?
Yes. something.
Both are heterozygote. One is with exon 1,
Mhm.
heterozygote, and one part of exon 3 is also heterozygote. Two
At the same-
double heterozygote, two compound heterozygote.
Mhm.
Heterozygote for exon 1,
Mhm.
heterozygote for exon 3.
Mhm.
Both of them are heterozygote but on the same
On the same locus.
gene.
Mhm.
We call them as compound heterozygote.
Mhm.
What's the double heterozygote?
Well, that's when you just add more alleles to it. I think.
One unit allele on
On two different locus. As I said, for example, we have different genes for some phenotypes. For example, here it does. One individual is heterozygote for a lupus on exon 1 and heterozygote for a lupus on a gene chromosome 1 and heterozygote for a gene which is located on chromosome 10. Okay? This is double heterozygote on two different lupus. Okay? But compound heterozygote is heterozygote for a single gene, but different part of these genes. Is it complicated or not? You understand me? So, compound heterozygote is two different mutations at a specific gene, but different part of these genes. Okay? Double heterozygote, two different mutations on two different genes on different locuses. What's the dominant and recessive alleles?
dominant is expressed and recessive is not expressed.
Dominant is expressed and recessive.
It's suppressed.
It's suppressed.
It is expressed, but later on. Like the recessive one, it will be expressed and it is present, but maybe like
The next
the
generation.
next generation.
What?
Like recessive genes, recessive alleles, they always tend to be suppressed in presence of another dominant one. So yes, that's it.
Do you mean the dominant allele is stronger allele?
Yeah, sort of.
You agree?
Its phenotype would be
If it is heterogeneous, then it is dominant. If it is homogeneous recessive, then recessive will show the phenotype. Like if capital R and small r, capital R will be the dominant. If both of them are smaller, then R.
it's the same thing. That in presence of the dominant allele, the recessive one will be always suppressed. Yeah, I mean,
All of you agree? Yeah. Let me have one example for you. Maybe you need to change your mind about this term. Okay, about the recessive or dominant, or about the dominant is more stronger than recessive. Maybe you need to change your mind about this. Okay. You have two copies. Okay, you have two copies for each genes, right? From father, one from our mother. Okay. One allele is mutated, or deleted. Okay? 50% of our protein is produced by this allele, 50% of our protein is produced by this one. Okay? One is deleted. Okay? We will have just 50% of the protein. Okay? So we have the phenotype. We will have disease. Okay? Right? So this is called as recessive or dominant.
Don't know. Might.
Government. Government.
If it's showing the phenotype, that
Let's
is.
say again, 50% of protein is produced by this allele, 50% of our protein is produced by this allele. One is from father, one is from mother. One allele is mutated, or one allele is deleted. Okay? This allele is active. It's produced 50% of our protein. But our phenotype is changed. We have some changes in our phenotype. We have some disease. Okay? What we call them? We call them as dominant or recessive.
Yes.
Since the disease is showing us, so we can see like
I think this is because the dominant is more.
I can't think.
It's dominant.
The mutate because the disease appears.
So, which one is stronger? This one
Yeah, that.
or this one? This is normal.
It's not exposed.
This is normal. 50% normal. 50% normal. 50% is deleted. Okay? Is deleted. 50% is deleted. We have phenotype. Okay? We call them as dominant. Which one is dominant? This one which is deleted, or this one which is not deleted?
No, I think this one.
This one?
Yes.
No, the deleted.
The deleted part? So when we talk about the dominant or recessive, what we talk about? We talk about the phenotype. Okay?
Yeah, okay.
Right?
Yeah.
I want to explain for you that dominant or recessive doesn't mean one allele is more stronger or the other allele is less strong.
It just means which
It
one
depends
exists.
on the,
The
The
phenotype.
one is in shoes, the one who expresses.
Which one is
it depends on the, our body's need to that protein. Okay? 50% of protein is produced by this allele. 50% of protein is produced by this allele. Okay? If our body needs 100% of this protein, okay, then one allele is deleted. Okay? We will have the phenotype. Why? Because our body needs 100% of this protein. 50% is deleted, then we will have changes in our phenotype. Okay? But if our body doesn't need 100% of that protein, we have many proteins in our body which we can tolerate. If 50% of protein is deleted, we will not have any phenotype. It means our body can work by 50% of protein. Okay, we call them as
Recessive.
recessive. Okay, because our body doesn't need 100% of that protein. Okay, so dominant and recessiveness is dependent on
Needs of the
our body's need to the protein. Okay.
So when
It doesn't mean this protein is stronger or this protein is not strong. Okay. So if our body needs 100% of the protein, if one allele is deleted, then we will have the phenotype, and then we call them as
Dominant.
dominant. But if our body needs to just 50% of a protein, okay, and one allele is deleted, our body can tolerate, we will not have any changes in our phenotype, then we call them as
Recessive.
recessive. Okay? So, dominant or recessive doesn't mean the allele, which allele is stronger or which is actually recessive.
Can you take it?
Okay. Some other terms. One is allele frequency. The frequency of an allele population, we call them as allele frequency. For example, if we have 50 individuals in a population, it means we have 100 alleles on that population. And when we talk about the frequency of allele, if, for example, among these alleles, 20 allele are capital R, the frequency of capital R allele in that population is 25% or 0.5. 0.25. And then we will have 75 small r in that population. Then the frequency of small r in that population is 75% or 0.75. The important note is the sum of the frequency of each allele in a population is 1 or is 100%. Okay? It means we can find the percentage of some allele on that population if we have the frequency of other alleles. Okay? Also, we have phenotypic or genotypic frequency and phenotypic frequency. The percentage of each genotype, each genotype in a population, we call them genotype frequency, and the frequency of each phenotype in a population, we call them phenotype frequency. I have collected some terms for you. We talk about some of them, and also you need to read by yourself in other terms if you read to know more. This is an important, actually, termination or terminology that I collected for you. We will have some exam questions for these terminologies. You need to know about them. As I said, we talk about some of them, and also if you want, you can study by yourself. You have a quick review on them, and then if you have any question, you can ask me. We talk about the allele, we talk about the autosomes, we talk about base pairing. What is base pairing?
And there is a pairing of different nitrogenous bases. For example,
When we
cytosine.
talk about DNA, we talk when we talk about the units of the DNA. Unit for the DNA is base pairing. This means the number of nucleotides. But pairing means, each nucleotide has a complementary nucleotide. A is complementary nucleotide for T, and C is complementary nucleotide for G. So we call them as base pairing. Okay? But our actual purpose is aiming at really the size of the DNA, the number of the nucleotide. Bases, each nucleotide in DNA carrier, an individual to carry a mutation or a genotype. Centromere, we will talk about them. Chromatin and chromosome, we will talk about them. Colon is a collection of genetically identical cells. Codon, the sequence of nucleotides, which code an amino acid. DNA sequence, dominance, we talked about it. Exons, we talked about the exon expression. What is expression? The transformation actually of the information. Gene, gene bank, collection of the DNA, gene technology. Gene therapy, we will talk about gene therapy, we will have a class of gene therapy. Genetic fingerprint, we will talk about it. The genome is the entire genetic information for a cell or an individual. Germline is actually cell sequence. Genome or sex chromosome, hemizygote, actually presence of a single gene or allele. Heterozygote or heterogeneity, we talked about it. Then hybridization, we will talk in other classes also. Intron, we talked about it. Phenotype, we will talk in other classes. Meiosis, messenger RNA, synthesis. Molecular genetic, we will talk. Mutation or alteration, we talked about it. Nucleotide, basic unit of DNA. PCR or polymerase chain reaction is a procedure that replicate the number of DNA in laboratory. When we want to have a genetic test, we collect the DNA from individual and then we amplify with DNA using the PCR technique, and then we can use this amplified DNA for sequencing and by identifying their mutation. The penetrance is the frequency of each allele, the phenotype. Actually, we talked about the phenotype, polygenic traits, is clear. Polymorphism, we talked about it. Protein, full base, limited base. Recessive, we talked about it. Replication, actually identical application of DNA. Sequencing of DNA, telomere, we talk about the telomere in cytogenetic class. Transcription, we talked about it. Translation also. You need to study these terms, and it's important for you to understand these terms, to understand the genetic, and also we will have some question of this term for the final exam. One important thing about the genetic is when you work as a practitioner, if you have some specialization in your field, if a patient comes to your office, maybe you have some medical test for he or she, you will have actually some family history, you take some history, and then you will have a diagnosis, and then you treat he or she will be treated. Okay, this is all you do in other field of the genetics. But in medical genetics, you need to know about the patient. You need to do all of this to have a diagnosis for that individual. But you need also to know about the disease. How this disease is transmitted. What's the pattern of inheritance? Okay? Not only about the patient, you need to know about the disease also. Because you are actually working or you are doing medical genetic specialist. Okay, you need to know about the disease, the pattern of inheritance, or kind of the pattern of inheritance. Is there any test for diagnosis of this genetic disorder? And also you need to know about the entire family. Okay, how many brothers and sisters they have, which individual in the family may be affected or may be carrier. So, in addition to the patient, you need to know about the disease and you need to know about the entire family to have a good actual practice in medical techniques. And then we divided the genetic disorders in three main categories. We will talk about them in next classes. One category is single gene disorder. It's clear the disease that causes by a single gene, changes in any single gene that can cause this disorder. We call them as a genetic single gene disease. Chromosomal disorders based on the chromosome deletion or replication of a whole chromosome or a part of a chromosome. We call them as chromosomal disorders. And the third part is complex or multifactorial or polygenic disorders. It's clear. There are, in addition to the genetic causes, there may be some non-genetic causes of this, some environmental factor, or not just a single gene. There are several genes that causes these diseases. We call them as polygenic or multifactorial. There is a website, it's called as OMI, Online Mendelian Inheritance in Men. It's a reference for single gene disorders. You can find this website at this address. There are more than actually 4,000 single gene disorders that actually there are information about them in this site. You can find the pattern of inheritance. You can find about the sign and symptoms of this single gene disorder in this website. You can find any laboratory or genetic test if there is for these disorders, and the pattern of inheritance and any information that we need to know about that single gene disorders. You can find in this website. As I said, more than 4,000 single gene disorders are located in this, are listed in this. Okay, this session of our class is finished. We will have another next session program in about around 10 o'clock.